Hello, and welcome to Biology 227. This is Anatomy and Physiology 1 for the College of Western Idaho. My name is Dr. Andy Jensen. I will be your instructor. All of the slide files will be provided via Blackboard, and of course the syllabus is always online. Most of your questions are answered in the syllabus, as far as which topics will be on an exam, the test days, and many other things. If you email me with a question that's answered in the syllabus, I will usually respond only with please reread the syllabus. So let's compare A and P just a little bit. We're going to describe the science of anatomy and physiology, and we're going to list subdivisions in anatomy and physiology. So here is a picture of the Edwin Smith Papyrus. This is one of the earliest known medical documents from about 3000 BC. It was apparently intended as a textbook on surgery. It begins with clinical cases of head injuries and works systematically down through the body, describing in detail examinations, diagnosis, and treatment, and prognosis of each case. It showed the ancient Egyptians' knowledge of the relationship between the pulse to the heart and how the stomach worked and large blood vessels. In 1543, Andreas Vesalius was the physician to the Pope, and he wrote a book called De Humane Corporis Fabrica. It was one of the first modern anatomy texts. In the 17th and 18th centuries, anatomy began to flourish. Attending a dissection was legal, although performing one was a little dicey. And the students of anatomy would have to travel from execution to execution because only criminals could be dissected. So after a criminal was executed, there would actually be bidding wars at the scaffold to obtain the body. In 1752, the Murder Act specified that anybody who had committed a murder shouldn't be buried. Everybody was buried in hollowed ground in graveyards, and that wasn't good for murderers. And so they were required to be dissected or gibbeted, which would be hung in a cage until the elements and animals had taken care of your body. Now, Scottish law determined that suitable corpses to undertake dissections were those who died in prison, suicide victims, and the bodies of foundlings and orphans. With the rise in prestige and popularity of medical training in Edinburgh, the legal supply of corpses could not keep up with the demand. Students and lecturers and grave robbers, also known as resurrection men, began an illicit trade in exhumed cadavers. In the 1820s, the residents of Edinburgh took to the streets to protest at the increase in grave robbing. To avoid corpses being disinterred, bereaved families could use several different techniques to deter the thieves. Guards could be hired to watch the graves. Watchtowers were built in several cemeteries. Large stone slabs could be placed over a grave for a short period until the body had begun to decay past the point of being useful. Or you could get a mort safe, which was an iron cage that surrounded the coffin. This high level of vigilance from the public and the techniques used to deter grave robbers led to what the historian Ruth Richardson described as a growing atmosphere of crisis among anatomists because of the shortage of corpses. This led finally to William Burke and William Hare, two Irishmen living in Edinburgh and Scotland, to commit 16 murders over the course of a year. And not only did they rob the bodies, but they sold the bodies then to the medical college that was in town. This caused a change called the Anatomy Act of 1832, which allowed doctors, teachers, and medical students to dissect donated bodies. So anatomy is going to examine the structure of the body parts. It's basically geography of the human body, but in Latin. And physiology examines how all those parts work. Pathology is when the physiology goes horribly wrong. The main theme of A and P is form fits function. For example, if you want to go play a basketball game, you don't wear 3-inch heels. If you want to go out for a night out of town, you probably don't wear scuffy basketball shoes. The function is separate, and so the form needs to change to fit the function. The scientific method refers to a systematic and rigorous process by which scientists examine natural events through observation and basically try to explain the world around us. Microscopic anatomy examines structures that cannot be observed by the unaided eye. So, in other words, through a microscope. Gross anatomy, or macroscopic anatomy, investigates structures visible to the unaided eye. It's not gross because it's disgusting. Gross means large and easy to see. If we look at a few divisions of microscopic anatomy, we have cytology, which is the study of cells, and histology, which is the study of tissues. In gross anatomy, our divisions are systemic anatomy, which studies anatomy through systems, and that's primarily what we're going to use in this class. There's also regional anatomy, studying anatomy by region, surface anatomy, comparative anatomy, which examines anatomical similarities between different species, and embryology, which studies developmental changes from conception to birth. Pathologic anatomy examines microscopic and macroscopic changes from disease, and radiographic anatomy investigates internal structures through the use of advanced imaging procedures. Physiologists can be general or specialized. They can look at the physiology of the cardiovascular system, neurophysiology, respiratory physiology, and so on. Pathophysiology is the relationship between the function of an organ system and disease or injury to that system. So as I mentioned, form and function are interrelated. So integrating anatomy and physiology is one of the easiest ways to learn both. In some colleges, classes of A and P are taught separately. First you learn the anatomy and then you learn the physiology. We like to do it combined here because we think it makes a little more sense. Now, what makes a living thing? There's a couple properties common to all organisms. One, they have to have some kind of organization. They have to describe some kind of structure. They have to have some kind of metabolism. They have to take in energy, and they have to use that energy to stay alive. They have to grow, they have to develop, they have to repair themselves. They have to respond to the environment. They have to have the ability to sense and react to stimuli. They also have to regulate their internal environment and the ability to reproduce. So if we go back one slide here and look at this, my car is organized. It's got a really complex structure. It takes in energy and burns that energy, but it doesn't grow. It doesn't develop. It is responsive. It can sense changes in speed and changes in temperature in the interior cabin and react to those stimuli. And it's able to regulate itself according to those senses. But again, it can't reproduce. And so it's missing out there. My car is not alive. So we've got 6 major characteristics of life: organization, metabolism, growth, responsiveness to the environment, regulation of the internal environment, and reproduction. There's several levels of organization when we look at living things. There's the chemical level, the cellular level, the tissue level, organ system, and organismal level. So let's look at the chemical level, which is the smallest. We've got atoms, which are the smallest units of matter, molecules as we stick atoms together, and then macromolecules as we make very large molecules. If we add enough macromolecules together, this will give us organelles. And then as we organize the organelles, we can get a cell, which is the smallest living structure. And we say it's living because it fits all 6 of the criteria that we've already talked about. As we move up to the tissue level, we have groups of cells performing similar functions. And then as we have different tissues coming together to perform functions, we have the organ level. We have 2 or more tissues performing specific functions working together. And then we have the organ system, where we've got several organs working together, and then the organismal level, which is the highest level, which is the entire organism, all of the organ systems working together. So in A&P, we're going to look at the integumentary system, the skeletal system, the muscular, the nervous, and the endocrine system. And that's where we will end with Biology 227. In 228, we'll start up with the cardiovascular system and move to the lymphatic system, the respiratory, the urinary, digestive, and reproductive systems. Now, to maintain life, we need water. Water is required for metabolism. It transports substances and it helps regulate body temperature. I want you to start thinking about the things I say in that, what do I mean by it's required for metabolism? What part of water is used? How is it used? So it turns out that metabolism is the use of chemicals to obtain energy to do the things that you need to do. To do that chemistry, the chemicals have to meet up with each other, and to do that, they have to be floating. There's no small elves inside of your cells pushing molecules together, and so they just have to randomly float around, and to do that, they have to float in something, and that something is water. Humans get their food from the chemicals we eat and nutrients. We use oxygen to help make energy. We use heat because heat will control the rate at which chemical reactions occur in our body. Too much heat, and we start damaging our chemicals. Too little heat, and those reactions can't happen at all. Imagine if you put a bread pan of dough into the oven but didn't turn the oven on. There's no chemistry that happens. And finally, pressure. We have both atmospheric and hydrostatic pressures. Now, homeostasis is an extremely critical idea in physiology. It's the body's ability to maintain an internal environment despite varying external conditions. You can see at the top here, this man in the blue is sitting in the sand in what looks like a desert. I would imagine it would be extremely hot there. Yet, what's his body temperature? It's 98.6 degrees Fahrenheit or 37 degrees Celsius. If you look at the little Eskimo kids at the bottom, they look to be standing on snow, wearing some coats. What's their internal body temperature? It's the same. And then over to the left, where I would like to be on the beach somewhere under a palm tree, what's that person's core temperature? So maintaining homeostasis requires most of our energy, and it's not a steady state. In other words, once we obtain our core temperature, we don't shut that system for maintaining that down. It's in constant dynamic equilibrium, meaning we have to keep on adjusting ourselves moment to moment to moment. One day you might be in the frozen north, another day you might be trekking through a desert, and your body has to change its responses in order to match the environment on the outside in order to maintain homeostasis. To do this, we've got three parts of a homeostatic system. There's a receptor, a control center, and an effector. Now, there's going to be some kind of parameter, some kind of variable that we have to sense in the surrounding environment. We have to detect some kind of change. So that's our receptor. Then our control center determines whether that change varies from what it should be. In the case of humans, that control center is the nervous system. We can also use the endocrine system for a more generalized and longer lasting response. And finally, the effector is the part of the body that will cause changes directed by the control center that will bring us back into homeostasis. So the most common way that we maintain homeostasis is by use of a negative feedback system. It controls almost all the processes in the body. There's variations that fluctuate from a normal range around a set point. If a stimulus increases, then the receptor informs the control system, which activates an effector to decrease the variable back into normal range, and the opposite if the stimulus decreases. In other words, we have a set point, and any change from that set point is minimized so that our measurement comes back to the set point. For example, temperature regulation. If you go travel to a cold area, your body temperature might start to drop. Sensor receptors in your skin and in your core are going to pick up on this temperature, and they don't make a judgment on the temperature. They simply send signals to the brain, and the brain, the hypothalamus, will then make the judgment on whether that temperature is high or low. If the temperature is low, as in the case we're describing here, then we're going to cause several changes to happen. You can have multiple effectors. So we're going to decrease blood flow to the surface of the body to help us not lose so much heat. We're also going to send impulses to skeletal muscles, which will then cause them to shiver, which will generate a lot of heat inside of the body. So this generation and retention of heat in the body helps to combat the cold temperature on the outside. As body temperature rises, then the sensory receptors are going to detect that. And again, they don't make a value judgment on that. They simply measure the change in the parameter, which in this case is temperature, and send a signal to the hypothalamus of the brain. The hypothalamus which is our control center, will then make a judgment, and in this case the temperature is too high inside of us, and so that's going to cause signals to be sent to the effectors to cause smooth muscle in the blood vessels of the skin to relax and to allow more blood into the skin to allow us to radiate heat away. It's the same thing with your furnace. If you set your temperature to 70 degrees, then your thermometer measures the current temperature in the house, and it's going to send a signal to the thermostat. Now, usually your thermometer is embedded within the thermostat, and so that kind of causes some confusion sometimes, but it's a separate part of the thermostat. So the thermometer measures the temperature, so that's a receptor, sends a signal to the control center, which is the thermostat. The thermostat determines whether the temperature has gone too low from the 70 degrees or too high. Say it's now 75 degrees, then the thermostat determines that we need to do something about that. So it's going to send signals to the effectors, which in this case would be your air conditioner. The air conditioner comes on and the temperature starts to come down. Now, the thermometer doesn't stop. It's continuously measuring temperature. As the temperature hits 70 degrees, that signal is sent to the control center. The thermostat determines that the proper temperature is reached, and our variance from the set point is now 0, and so the signal to the effector stops. Cruise control in a car is the same way. In this case, the parameter we're measuring is speed. The receptor would be the speedometer, and the control center would be the computer in your car or your cruise control. If you start to go down a hill and your car starts to roll a little bit faster, then that's picked up by the speedometer, sent to the cruise control, and the cruise control says, hey, we're going a little fast here, and stop sending so much gas to the engine. So in this case, the engine or the brakes are going to be the effector. Say your car is going up a hill, the speedometer now sends a speed signal to the cruise control, and the cruise control determines that we're going a little bit slow. It's then going to send a signal to the engine to apply a little more gas so that we go back up to the speed that we set on the cruise control. So this is a negative feedback system. We're trying to get back to the point that we set. So the negative means that we're trying to minimize any change from that set point or set range. Again, here's the example of core temperature. So as core temperature starts to drop, that's picked up by receptors sent to the hypothalamus, which is the control center, and then the control center is going to send signals to the effectors, which are going to be skeletal muscles to cause shivering and blood vessels to the skin to prevent heat loss. On the other hand, if the temperature is hot, that's picked up by receptors, sent to the control center in the hypothalamus again, and we're going to use some different effectors this time. We're going to increase blood flow to the skin and also activate sweat glands. Now remember, our receptors, the things that are measuring the temperature, never stop. They're continuously measuring temperature, continuously sending signals to the control center, which is continuously making judgments about whether that change in parameter has gone too high or too low, and then what to do about it to bring it back to where it needs to be. So again, negative feedback systems are the most common type of feedback system in the body. Examples would be things like temperature regulation, breath holding, the knee-jerk reflex. The list goes on and on. Now, positive feedback is where we're going to take and magnify the change from the set point. So it seems a little weird that we would want to do that, but positive feedback systems are really good for getting a single job done and then stopping. Negative feedback systems are really good for maintaining the status quo for decades. I mean, you've been pretty good at maintaining your body temperature for as long as you've been alive. So with positive feedback, one example here is going to be uterine contractions in labor. As the baby's head pushes against the cervix, which is an internal anatomic structure in a female, the cervix then sends stretch signals up to the brain. The brain then releases, in this case, oxytocin, which causes the uterus to contract harder which in turn pushes the baby's head against the cervix even more, causing it to stretch even more, causing more signals to go to the brain to say, hey, it's being stretched, which then causes the brain to make more oxytocin, which in turn causes the uterus to contract more, and it just keeps magnifying and cycling and getting more and more and more extreme. And the idea here is that you want to deliver the baby. You don't want to maintain the status quo and hold the baby in the birth canal for decades. Another example of positive feedback would be breastfeeding. So as the baby suckles, the receptors in the nipple pick that up, send that signal to the hypothalamus, which is our control center, and then it's going to send signals to the posterior pituitary, oxytocin is released, and breast milk is ejected. And this will keep going as long as the baby continues to suckle. Now, as the baby stops suckling, the receptors stop sending that signal to the control center, and so oxytocin will stop being released. And it takes a few minutes for this cycle to kind of wind down here, but as long as the baby continues to suckle, that positive feedback will keep the milk being expressed. Interestingly enough, a lot of people don't know that milk is expressed under pressure from the breast. When you look at a newborn infant, they're tiny. They're really, really tiny. They don't have the sucking capacity to pull milk out of the breast. So instead, as they suckle, the breast pushes the milk into them. So with both positive and negative feedback systems, they're both ways to maintain homeostasis. It's a dynamic equilibrium. We're constantly measuring any given parameter, making decisions on that parameter, and then sending signals to effectors to help bring our internal environment back into balance. We have a receptor, a control center, and an effector. If homeostasis fails, that's where disease happens. So you don't need to memorize these here, but for example, some parameters of normal for homeostasis. So these would be for negative feedback systems, would be 98.6 degrees Fahrenheit, 80 to 110 milligrams per deciliter blood glucose, blood pressure of 120 over 80. And we got these numbers by looking at lots and lots and lots of people that were deemed to be healthy, measuring their parameter, and then using some statistics. So the normal range for this value is normal for 95% of people. Now, 5% of the healthy people we looked at had values outside of this normal range. So this is where being a physician comes in. You have to understand that sometimes a patient can have a body temperature slightly higher or slightly lower, and that's normal for them. So diabetes would be an example of a homeostatic imbalance. So if we can't regulate blood sugar or blood glucose, then that's going to cause some issues. To treat any disease, we first have to make a diagnosis, find the specific cause of the homeostatic imbalance. Now, if we can correct the cause of the imbalance, then we can do that and the patient will be cured. Unfortunately, a lot of times that can't happen, and so most medications are going to kind of grab the control center by the horns and force it in one direction or another. So as a clinician or a physician, as you develop a diagnosis, you're going to use that scientific method. You examine the patient, you gather data, you come up with an initial hypothesis. You order some tests, which would be the experiment in the science here, and then you're going to confirm, modify, or reject your initial hypothesis or diagnosis, and then keep repeating this until you have a confirmation and a definitive diagnosis. And then once you have that, you're able to treat the patient. Now, as I mentioned, drugs are going to affect normal homeostatic control mechanisms kind of by grabbing them by the horns. So if a patient has lower levels of serotonin in their brain, we can give them a drug called an SSRI, which will bring that back up to the normal range. Now, if you give somebody who's already in the normal range an SSRI, it will boost their serotonin levels up past where normal is into the high range.